So the thing that we'll be mostly talking about is the proteomics and the interactomics, which is to understand the interactions using the high throughput arrays. So basically, so today we will be mostly looking into it. What are the different technologies or the techniques that are available for the uh, proteomics, and as well as what are the different high throughput array techniques that are present and for understanding the molecular levels and the systems level uh, biology. So moving on, so also what are the different concepts, and what are the how different advantages and disadvantages for using each of those technique, and how all those things can be taken forward, and how can be to understand the systems at a better level. So firstly, before moving on to the exactly the concepts of the omics technologies, about the proteomics and the interactomics technologies. So basically, before moving on, so what is actually the concept of the proteomics, and what is from where it has evolved, and how it has we have gone through. So to understand it, we can understand in a way that uh, if we look at the central dogma and how the things have evolved, right from the genes to the genomes, genes, then the DNA, then gets transcribed to RNA, and then finally we get the proteins, and then finally we get the metabolites. But the what is the meaning of this term omics, or what from where the things came or evolved? So the basically the study of everything is comprises of omics, like the study of all genes, which comprises of your genomics. So the it all started, that with with the starting of the human genome project, which was very much successful, and we got to know about the sequences of all the genes present in a human. But the major thing that remains still challenge is that it was not completely all the information that we were looking for could not be comprised of the human genome project could not answer all those questions. So for example, the gene sequences or the sequences, if we got or if we have analyzed as well, but the major problem, you can see that mostly all the organisms, as well as during the disease, most of the organisms, only one, when found out that all the genomes were analyzed, could found out that the sequences of most of the organisms are quite similar, but the structure and function and the behavioral changes are manifold for each of the individual organism. So obviously the question that came into mind, that genomes or the genes through which the sequences we can get an idea of various particular things, like the sequences, what are the mutations are there, but mostly the genes are quite static in nature, and which gets further transcribed into the RNA, and which is then only further responsible for translating it into the protein or the functional molecule, which leads to the changes and leads to the understanding of the a person and a human being. So basically, moving on to understand that the omics technologies, so only through the genome sequences of the genome project, it won't be able to completely answer all the questions. Rather, we should look into the at the next level, which comprises of your understanding the gene expression level, or the understanding all the transcripts in a particular human being, be it in your particular cell or particular organism, or particular in a diseased condition, or those things. So in the field known as your transcriptomics, or else what we can say that in the further moving on is the proteins, which are the functional molecules and are responsible for the human beings, for the persons to actually work on uh, or work on this or particular thing. So that's why it is the further studying of all the proteins in that organism is the field of proteomics which emerged, and also the field keeps on emerging with the studying of the functional molecules like metabolites and the metabolomics. But mostly what we focusing on the different applications of the proteomics technologies, and how the field has evolved and has given answer to many concepts, and also the different interactions, the not only the expression level of the proteins, but the interactions also play a huge role, and how to study specifically those interactions, and how we can get a another level of understanding for in a better way. So this is an overview of the complete omics-based technologies. And now, the as I mentioned that the genomics and the gene, the after the Human Genome Project and the genomes and the genes. So now the question came that how the proteomics field become very much useful or became very much needed. So definitely the proteomics. So the question that how this proteomics will become so much indispensable in this things to for the study. So the genome sequences failed to answer all the questions, as I mentioned. So with the help of this central dogma and this thing, we can easily understand that whatever we get, finally the functional molecules remains the proteins. So that's why it helps us in addressing many questions or many things which cannot be answered by the at the transcript level or the gene level, can be answered by those things. Now for example, so if we take in a as a very good example. For example, in the tumors, for example, in the diseases like cancer and tumors, so there are different kinds of therapies that are being used. So so everyone is well aware. I hope that everyone is well aware that giving the drugs and the chemotherapeutic drugs so always does not work on each and every person, and that mostly that can be due to the way the drug acts. So the if the receptors are not present on a particular or particular individual, then that drug might not bind and might not work. As well as the if for example the immunotherapy, which are very much being on the upfront and on the rise, but still the immunotherapies will work on that will depend on much another things with the how the immune landscape of that individual is, or how that works on that, and the surgery and all remains the basic major things. But now the thing is that which is coming is the personalized therapy, which is the one of the most upcoming thing in the near future. So this is because the major thing for the personalized therapy, the major concept remains is that to have a right patient, right drug, and the right dose. So which patient needs to be administered with which drug, and uh, at which dose is the major question that needs to be answered. But the major so how to answer those? So we all know that certain questions can be answered. Uh, so certain questions can be answered with a particular molecule, with a particular like the gene sequence, or the that receptor or the genomic receptor is present or not. But always the molecule cannot be, as we already understood that the proteins or the functional protein molecules are there, which needs to be answered and which needs to be understood. So that's why it proteomics can play a huge role in understanding or in the field of personalized therapy, where we can understand firstly through the protein biomarkers, which we can understand the early diagnosis that whether the patient is being is going to have that disease or not, to so diagnose that patient. Second is then the patient outcome. So if that protein is overexpressed in that patient, then what if the drug is administered, then whether the so how much the drug will be effective on that patient. as well as the disease progression, right from that how the diseases can be progressed. So if the patient is going to in a very high grade stage or the metastatic stage or not, so all these things can be addressed by the or can be answered by the proteomics field of proteomics. So the proteomics questions plays a huge role, which helps us to understand in multifaceted way. And in this era or this upcoming era of personalized therapy, if we say, then definitely the major thing that will be playing the role is for the completely regarding the proteomics will play a huge major chunk of this role. So that's why knowing the technologies, knowing the upcoming technologies, knowing how to uh, get or to answer or to understand that how all the protein sequences that we are getting. So that is that how the all the proteins in an organism in a disease state or in a normal state, how to get their expression levels, how to identify all those proteins. So for that studying of this technology and of this uh, becomes very much important and necessary. So now how the evolution, so the major thing is that, so we know that this complete field which got boosted up with the genome sequencing and to get the human genome project rolling. But similarly in this about the proteomics technologies to identify and to get information of all the proteins in a human being or in an organism. So it also got evolved to various stages, and basically to say it is still evolving with the newer technologies, and specifically the coming up of the technologies like the at the understanding at the single cell level. So now all these technologies that we are talking about is at happening at our molecular level, but the field has traversed and is now being advancing towards to understand at the single cellular level with single cell RNA-seq or the single cell proteomics technologies. So we can understand that the field is still evolving, but still we can get a basic understanding that how the fields have emerged and from where. So the how from the different throughput techniques like the before how the was very much was very much restricted to certain level of throughput, but now are continuously evolving and being going to the very high throughput techniques. So in the if you so in 1975, so O'Farrell is the one who developed this concept of the 2D electrophoresis, which we will also look in somewhat detail in the upcoming slide. So he was the one who which tried to understand and find the protein patterns and the protein interaction patterns and the difference in the proteins, and tried to separate the proteins on the basis of the two different dimensions and to find out. But it was, you can understand that it was not only very much labor intensive, which was very much labor intensive, then it was not so much effective and not so much high throughput. And also the you can understand that all the proteins cannot be just identified, and the expression level getting the expression level was not so easy or not so relevant during that time. So now moving on the field as it continued to evolve. So till that time also there was the term proteome was not there in the literature, or it was not being used by the researchers at that time or by the science community. So it was in the year only 1995 when Dr. Mark Wilkins has coined the term proteome to let understand that what is to let the scientific community know to have the all the proteins that he had studied in east, that to say that to refer to that actually refer to that phenomena where he wanted to express that he had studied the almost all the proteins in the east, so has coined the term proteome. And so basically, so the ohm generally comes, and the meaning is that to have the study of all. So like the genome having the study of all genes. So similarly, the term proteome came from that to study of all the proteins. So now after now in that time, so another thing that emerged up was during the 1980s and 2000s was the ionization techniques, and to have the mass spectrometry based approaches. So advancement in the technique, one can try to identify as many as proteins. But the major challenge, and then further moving on to the targeted proteomics, so we will look into that. So not only the discovery phase of the proteins or the proteomics field, so to understand and analyze and identify different protein levels, but also to validate the proteins, which is very much only dependent on certain antibodies and all those. But in the year 2012 also we could see that we can get the field, and we can get through mass spectrometry based approaches only we can validate also certain proteins. So the all these things, all these approaches which we saw, or the advancement which we could see, was only possible because of two of the scientists who could got the way to ionize the proteins without denaturing those, and to get the ions in a manner that the which is also known as your soft ionization, and also the invention of the electrospray ionization techniques. So this only helped us to get these advancements where where we can identify a lot of proteins, a lot of uh, biomolecules, and also can validate those. But this could not be possible because the laser ionization desorption method that was present was mostly being used at a level where the biomolecules or the chemical molecules are being analyzed, where the disruption of that things the disruption happens, and it does not affect so much. But the protein being a biomolecule, and to have those intact, it was very much needed to have a controlled ionization. And without the ionization, it was not being possible in the mass spectrometer, because it is the ions that gets detected in the different after getting separated in different mass analyzers and getting detected in the ion detector. So that creation of that ions in a controlled manner and have an electrospray ionization techniques are the which also fetched gave them the Nobel Prize. So this made the field very much advanced, and this facilitated the actual growth of this field to the uh, very much advanced level. So now, after the techniques have been advanced, and the way we had the human genome map, the human genome project. So similarly, with the advancement of these mass spectrometry based techniques, and the uh, with the as from the ionization techniques we got the mass spectrometers, and then there was a huge change in the mass spectrometer with the different mass analyzers being used to have a better peptide separation and the better ion separation, and to get detect as many as ions possible with a higher resolution and higher uh, scan ability, and then finally detecting those ions, and then with the evolution of the different algorithms. So because once the thing that generally gets detected are the ions, but from that ions how to calculate back your peptides, and then finally the proteins. So then in 2014 was the draft map of the human proteome came, which identified around your 85 or 84% of the total annotated protein coding genes. But you can understand that among the 20,000 or the 20,000 or odd those genes present, only 84% of the total protein coding genes
இன்று எனது வாழ்வில் மிக துன்பமான நாள். ஒரு
was
தந்தையாக இருந்தும் தன் புதல்வரின் அறைகூவலை எதிர்த்து யுத்த பூமியில் और पूरी YouTube
Is
बनते जन्नत
everyone mute?
Yeah, so only this only 84% of those proteins could only be analyzed, could only be identified, and still 16% could not be done. So these reasons can be many. One thing can be the because of the uh, uh, that's low abundant peptides and the low expression level of the proteins which does not get detected, as well as the instruments have certain limitations, which one option can be that. Another thing is that the proteins are not expressed at that individual of all the what all the individual that was taken for the particular uh, that time. So since the proteins remains vary and also are very much dynamic in nature, so it sometimes all the proteins does not express at a particular time in a human being. So with that the proteins cannot be could not be identified. The reasons can vary, and you can think of as many possible reasons possible for that, but still could identified around 84% of the total annotated protein coding genes with wide range of expression across the tissues. So now is the next in the after the mass spectrometry based approach of the human proteome. So another came up with the antibody based approach. So you can understand that the major difference and major thing between a mass spectrometry based and antibody based methods are that the antibody based methods require the particular antibodies and have a kind of a cost associated with it, but it is kind of a much robust in a way. So this in 2015 not only they did an RNA seq or the RNA or the gene level expression of the tissues of the particular genes at in the different organs, also the uh, of the across the different 32 human tissues, what is the protein level expression or protein level profiling, giving another comprehensive proteome map and the human protein atlas. So now moving on from 2014, 2015, then in 2020 with the Human Proteome Organization. So Human Proteome Organization, so just give you a definition. So Human Proteome Organization is something, is an organization which has been named and collaborated with different institutes and different countries to facilitate the proteomics research across the globe. So they had launched a project in 2010 for the Human Proteome Project, and which in 2020 they had published that they could identify around 90.4% of the total human proteins. And so this also marked the 10th anniversary of the Human Proteome Project, and which showed that around 90.4% of the proteins finally could be identified. So you can understand that moving on the field with, because of the evolution of different technologies and the evolution of the different fields. So this was very much needed, and this was very much so with the evolution of different technologies, the mass spectrometry technologies. So what it happened is that one could got a very uh, best understanding and could identify a lot of proteins, but still 10% could not be identified, which are known as your some of your are missing proteins. So for example the proteins could had so there are different levels of proteins that are there, like P1 level protein, P2 protein, or P3 protein. So some proteins are evidenced at the transcript level, some proteins are evidenced at the have been identified through antibody-based method, but could not be identified through mass spectrometry-based method, and etc. etc. So these are the different P status proteins are there. So some are known as your missing proteins. So these missing proteins are the ones yet to be deciphered in this field, yet to be known that these proteins, what is the how what is the expression level of these particular proteins in different tissues. So this gives us again a scope or a chance to explore in this particular field. Also, the missing proteins, the reasons again can be many. So not only the with the limitation of the instruments and getting a very low abundant peptides and proteins is a limitation. Also, it can be that the proteins remains absent in a normal individual, or for example, the fetal proteins will not get in an adult tissue, and like those things, which remains a challenge in getting all those proteins, particular proteins. So now, once we have got the set and understood that what are the different fields of this particular proteins and this proteome, this field with the evolution, the major goal was to get the soft ionization techniques and to get majorly the chunk of the challenge was to have ionization techniques. So now next on, and then the draft map of the human proteome, and we can see that it was in 2020 only, just by this 90% of the proteins, annotated proteins, could be published. So now the proteomics has a wide varied of applications and not limited to just any one or two. As we know that in the central dogma, this proteomics is not only the ones which is the proteins are the not only the ones which facilitates our particular not only facilitates the functional biomolecule and plays a role, and which is very much dynamic, but also the post translational modifications and all those things also plays a very much uh, plays a huge role in particularly in arranging, in maintaining signal transduction, enzyme cascades, and various other things. So studying PTMs and the post translational modifications also gives us a huge kind of it gives a huge kind of uh, uh, understanding that what is happening at the cellular, uh, and gives us a mechanistic insights at the cellular levels and the cellular machineries, that how that particular things is moving. And also the PTMs are not very few ones, not a very few. So there's a whole levels of PTMs, for example, phosphorylation, glycosylation, butylation, acetylation, and methylation, and name to you can name as many as you can. So understanding those particular things, that at uh, a particular disease condition, in that particular phosphorylation is happening or not happening, and what is its effect in the developmental process, in the growth process, in the disease condition, varying from organism to organism. So what is the particular things? So it's a huge role. So proteomics, one of the major field under proteomics, comes under post translation modifications, which helps you to get that knowledge, or to get that understanding of all the uh, status for the PTM. So this is also a very much emerging field and very much field to explore for to get a better understanding. Next, for the protein protein interaction. So now, if we just think of that the proteins that play a role, so these proteins not so, again, if you go back in the central dogma and find out that the proteins are the ones which are playing a huge role in building functional molecules and the functions of the organism. But this is not only that one particular thing or one particular condition. The major thing is that the proteins also interact with other biomolecules, as well as protein interacts with another protein, which leads this to the functionalities, or the having another functionality, or having certain effect, and can have a downstream cascades of effects. So it's always need to understand. So once we have identified proteins, so again we need to have an understanding that what is the way the proteins are interacting. So that's what we will look to some aspect today, that how we can study those interactions. So the significance, I think I don't need to stress upon the fact of the importance of understanding the protein-protein interactions, because this is a cascade of events that can be either upregulated or downregulated, or can be initiated, or can be activated, or uh, can be stopped using this interaction. So there are some of the methods like is to hybrid, co-precipitation, immunoprecipitation, microarray technology. So all this comes under the broad field of proteomics to understand the protein-protein interactions, also the protein drug interactions, or the protein DNA interactions, or what will be the effect if you treat any organism or anything. So all this comes under the field of protein-protein interactions. Next is the structural proteomics. So again, the we all know that the protein structure is plays a huge role to have the structural integrity for its original functionality, and to have a very good functional method. So the thing is that the protein so the have the protein complexes, how it is cross-linked with different at different locations, then the how to have the understanding of the when the proteins are for example, if an antigen and antibodies interacting, then how the structural conformities are there, and how the changes are being happening. So all this comes under your field of structural proteomics. Sorry. So now again going for the functional proteomics. So not only the structural, now to understand that how, so if you have a very uncharacterized protein, or the protein which is not very much characterized, or the roles of those particular proteins, so how to have those how to know that, so that can be known through functional proteomics. So one way is to indirect way is to have a protein protein interactions, through which we can understand that the protein is playing role in which part of the family of the in which families part, and through that we can understand that how the protein protein interactions can take place. Second is the elucidating the molecular mechanisms, that in that particular pathway where it is playing the role, or how it is working. So this comes under your topic for your functional proteomics. Next is the proteome mining, so which says that about your to have the to identify your target. So which drugs, if you want to administer, if you want to design certain drugs against a particular disease, and if you want to have particular drugs against those that, so for that we need to have a target identification. So as on the personalized medicine, I have mentioned that we need to have the known that particular drug is working against which particular biomolecule. Now the major thing after to, if you are not blocking any transcription factor, or if you are not blocking any particular gene, or not blocking any particular marker, if you are your drug is working against a particular protein, or trying to downregulate a particular enzyme cascade, so definitely you need to have the particular targets identified, that how relevant it is for your particular disease, how relevant it is for that particular individual to be administered to for that drug, further leading to the drug discovery, and all those which comes under your proteome mining. And another major major important is the protein expression. So this gives us a very good cue of the or the understanding of how the particular human being or particular uh, disease condition, how the proteins are getting upregulated, downregulated, how the expression levels are there, then to have one better understanding of the signaling cascades, etc. So all this gives under the comes under the protein expression. So the major so proteomics is not limited to only identification of proteins. So if you feel, or if you have an feeling that the proteomics is something that remains confined to just an identification of particular proteins, and to find out what are their what is the proteins expression levels. So this is just one part of the field of the proteomics, broad field of proteomics. There is just the protein expression profiling or the expression profiling, which is again very much important and very much needed for the understanding at in any disease condition to have a better view that how if the disease is person is progressing, that can be of any nature of disease, that can be your any infectious disease, like you know that there was huge need to understand in infectious diseases as well, that whether a person will be going to the severity or not, whether a person will become severe or not, again that a particular vaccine will work or not, or the epitopes or those things are getting changed or not, and many more things, and what are the particular expression levels and how the immune system is working. But the so that comes under one field, but others are equally if not more but equally important, like the PTMs, understanding the PPIs, then the structural proteomics and the functional proteomics, and then finally the proteome mining is also plays a huge role, and specifically in this upcoming era of the personalized medicine. So now, if we look into the proteomics technologies, so as we mentioned that the field has evolved quite a long time from the 2D gel electrophoresis to the ionization techniques and the mass spectrometers with the very high end mass analyzers. So obviously, what are the so we will discuss in a few uh, bit about all those things. So how what are the different technologies that are there? So one is always the gel based proteomics. So gel based proteomics is something which includes like the 2D gel electrophoresis, 2D dyes, but the major problem of the chunk is that it is definitely low throughput in nature. So the it started from here, and is also a very good and very robust way to have an understanding that at a single go, if you just profile the protein of any organism or of any of any proteins you take, you just give it in the you just run in the jail and you got the protein profile, and then try to find out the match in the differences, what is the way a protein profile of an for example X is differing from Y, but it is low throughput in nature. Then one is the mass spectrometry based proteomics, which includes many techniques like the level free quantification technologies or the label based quantification, then the targeted proteomics. And the major advantage of this are the it is high throughput in nature, which is very much helpful in getting an overview of all the mechanistic insights and the mechanistic use of the particular thing. The next is the antibody-based approaches, which includes your immunohistochemistry, immunofluorescence, Western blot, ELISA. Again, these are low throughput in nature. Not only low throughput, these are labor intensive. Second is the very much not so much cost effective. You need to have an antibody. You need to raise an antibody against your particular protein that you are being targeting, and then only you can perform the experiment, and then you can only say that you are getting the final output or the final result, that whether that particular protein is present or not, or how much is present. So Western blot, ELISA, but still these are some of the gold standards that are being still being used by labs, clinics, etc., like the ELISA, and many confirmatory tests for different for different diseases still is being done on Western blot and ELISA, but this is the techniques are itself low throughput in nature with varied constraints that needs to be done. So now again, if we divide it, this thing, the field of proteomics. So these are the technologies. One thing is the technologies that the gel based or mass spectrometry based or the antibody based, but another through which we can divide it into is that the phase of the study. So if you have taken, for example, a new you have started a new work on a particular disease. It can be any disease of the choice. So now the major thing is that whenever, for example, you have taken the protein samples or the biome specimens or bioanalytes from a particular patient sample and did adjust the profiling of the in the gel using 2D gel electrophoresis or the 2D dye for that matter. So you are just checking the profile without having any idea of which particular protein yeah, or which particular profile or at which particular spot in the gel might be changed or might be disrupted. You have no such idea. You are just going by the just looking at the protein profile. So that gives us the discovery phase. Similarly, for the mass spectrometry based approaches, so when we are saying that we are just taking the sample of the patient and sample and we're just running in the mass spectrometry and trying to identify that whether that particular what are the different proteins that have been identified in that particular patient and what are the how what is their quantitation levels or what are their levels of the expression levels. So again we don't have any idea about that particular protein. So now when we talk about in general antibody based approaches, so you need so whenever we are saying antibody, so we are having an idea against the of the antibody against which a particular protein antibody is raised against which a particular protein, otherwise whom to target or how we can target a particular uh, organism or particular protein. So for that reason the antibody whenever we are using, so we know we have an idea about that particular protein or which particular protein we are working in. So that by nature or by default comes under your a kind of validation phase where also like ELISA is being used for validatory or confirmatory results for many in the clinical settings for many diseases. So this again comes under the validation phase. Whereas in the mass spectrometry based proteomics, targeted proteomics is something which can be taken or considered as a validation phase because you know which particular proteins or you have an idea or mind or in mind as well as in the list of proteins that you are trying to uh, verify and try to find out in the mass what is their expression level across your samples. So in a for the validation phase. But overall, in the gel based and in the global mass spectrometry based techniques, when you are trying to characterize all the proteins and all the protein profiles, then definitely this comprises of your uh, in the discovery phase. Okay, so these are the technologies and the phases through which it can be divided and can be considered for. Now in the gel based proteomics, so we'll just get in brief about all the techniques, all the technologies, that how what is their basic principle and how they are done, so that we can also meanwhile we can understand that how the thinking and of the scientists and of the people in this field and the community have changed, and how one can and how the one can get the benefit of further using different high throughput technologies. So the which started around 1970s, the 1975 of the 2D electrophoresis. So this was simple concept. So the SDS page gel or the where the in the SDS page, the separation is done only on the basis of the molecular weight. So now the it is very much general to understand that many proteins in an organism can have will have the same molecular weight. Now, if you try to uh, separate it on the basis of only molecular weight, then it becomes very difficult. There bands and the, the bands won't be contain a whole lot of proteins, and it won't be that all the proteins are very much resolved. It won't be the case. So because you need to, if you are resolving in a standard gel, it won't be possible for you to resolve all the gels in the gel all the proteins, and it will in the bands there will be many proteins, and we cannot understand their profile. The simple logic was to divide it into in a two dimension, that where first dimension will be used uh, for division or dividing the proteins in through another uh, to their another property, and that property was taken was the isoelectric point, and was divided into and was first run in a IPG strip, which is having a low pH, having a pH gradient or the pH range, so that the proteins first will get divided into their isoelectric point, because in that isoelectric point the thing that will happen is that the proteins, their net charge will be zero and the proteins won't move there. And once the proteins got fixed on a particular first dimension at that IPG strip, and then when it is dissolved into the molecular weight, so then you will be able to dissolve it. So here in this example you can see that both A and C had same isoelectric point but had different molecular weight. So firstly, different pH gradient strip uh, has been separated, and though they had the same uh, pi or the isoelectric point, but since they had different molecular weights, when they were run in the second dimension, they could be separated. So this given added advantage or added edge over just the single dimension or just one dimension of the uh, of one dimension where separation was done. So it's just separating on a particular dimension or the IPG strip won't be possible, or just separation on this particular molecular weight, because many proteins will have also similar molecular weight but might be different isoelectric point where they will be divided on that pH strip. So that's what gave an added edge to have a better profiling and better separation and the dissolving of the protein spots. And then another technique which has been used very much specifically to analyze to analyze and to understand in a diseased condition, and to compare basically to in a quantitative nature. So this mostly you can think of in a kind of a qualitative nature, but when we talk about 2D dyes or the differential in gel electrophoresis, that you can think of or you can consider of something of a kind of quantitative in nature. So the meaning of the word where I try to express quantitative in nature is that the quantitative means that the you can compare the expression level in the two different conditions in two different things, so which and through the gel based approach, so which also gives us an edge over simple 2D electrophoresis, because uh, always whenever we are trying to compare this kind of thing of two different expression levels or the two different conditions, what is there and what is being happening. So always it is good, and it is good to have always to have a quantitative differences to identify and have a quantitative differences. So here what happens is that the samples, if you take and the protein samples from two different persons, so generally it is done with the help of fluorescence dye. So two types of dyes are used in two different samples. So for example here it has been beautifully given like the condition A and the condition B, you are just taking two different dyes. One is psi 3 and another is psi 5 dye. Whereas you just take the pool of these two and do with a psi 2 dye. Then what we do is that we all the three samples are run in a gel, and the major thing is that since we are utilizing different dyes like the psi 3 dye, the psi 5 dye, and the psi 2 dye, so whenever we do fluorescence or the fluorescence we measure at the different intensities, where the psi 3 dye at which nanometer the psi 3 dye gets excited, at which at the different nanometer the psi 5 dye gets excited, and that at different dye the psi 2 will be getting excited. So this gives us an image of the three different gels. So at the three different intensity at different excitation wavelengths. So we can get the image of the three samples, like the sample condition A, sample condition B, and the sample condition A and B. There is basically the pool. So this, the red and the blue, the psi 3 and the psi 5, we can compare the spots to find out that okay, at which particular this particular protein, which particular proteins are actually getting differentiated or getting the differences, or what are the difference level of those particular proteins. And this pool is nothing just to normalize and to have a reference point if the protein spot and the spots are getting differentiated, are getting differences due to the run to run variations and to handling and the technical variations. To reduce that, we are getting that particular gel bar. Rest the this helps us to get the conditions in a very fruitful way to compare the two conditions. Now, after the gel-based proteomics, the thing that we mostly is used in the field and is very much uh, helpful in to get the maximum coverage and to identify a huge number of proteins, and finally not only helped in contributing to get the human proteome map, but also different organs map like the human heart proteome map or the human brain proteome map. So this helps us also to get an idea, overall idea, or the profile of a particular protein expression in an individual or in an organism whenever in different conditions. So if we see that this is a simple three-step procedure to get analyzed, or the peptides to be get detected in a mass spectrometer, and finally get the, with the help of different algorithms and different analysis techniques and the different software, we can get to know and to have an idea about what are the proteins and what are their intensity levels. So the basically the first step is to have a peptide separation. So for this, chromatographic based techniques are used. So since the peptides are huge in number, so the thing is that it, if we just bombard all the peptides at once to the instrument, there will be a lot many number of ions which cannot get detected at the same time, and the detector will get saturated, and there will be a huge loss in the ions and loss in the understanding of the peptides. So the peptides needs to be separated through a liquid chromatography, and here which mostly is being used is the reverse phase chromatography using a hydrophobic material like C18, where the peptides gets eluted out, and after elution of the peptides through the help of ionization, so these peptides are uh, created into the ions, which goes into the mass analyzers, and where in the mass analyzers again ions gets separated, and finally in the detector those are separated. So basically it is the m by z's, or mass per charge ratio, that is being measured by the mass spectrometry. So once we get the uh, charges, the mass per charges ratio, and which will have different for different ions, then that charge mass per charge ratio can be taken and can be looked at the can be looked for different ion spectra for the particular peptides and particular ions, and which can be further taken forward and can be used for the for our analysis. So this is basically the concept of how the mass spectrometer works with three different phases, and the major thing that gets measured is the m by z charge for the particular ions. Now, the thing that I have mentioned or I started is, if you have noticed and if you haven't noticed, so please notice now. So that we started with the peptide, but if you remember and if you recall, we are talking about the proteins or the field of proteomics about the field of proteins. So we were talking about we were talking about proteins, but now we here also we had talked about the proteins on the basis of the molecular weight and ions, but here we had started talking about the peptide. So we haven't spoken about, or we haven't talked about proteins as such, but rather peptides. So what is that? So the major thing or the major field that happens is that there are two approaches, major approaches in the field of proteomics. One is the top-down approach, another is a bottom-up approach. So whenever the major advancement or the major most popular or the most thing that is being generally used is the bottom-up approach. So what we talked about just that the peptides. So this comes under the bottom-up approach. So what is so before going to that? So what is the two methods and how just two are different? So what is the top-down proteomics? So top-down proteomics is to have an measurement of an intact protein. So where we have the entire protein and the molecular mass database is being searched for that identification of proteins, and the MSMS fragmentation of an intact ion happens, where the intact ion gets separated to and have the sequence specific ion search. So here the basically the meaning is to have the whole protein and to without breaking that protein. So if you want to have it in a native state, and not in a that meaning, but to have the entire protein intact mass getting analyzed. So that comes under the top down proteomics, but the thing that is being used is the bottom up proteomics, which is where the proteins are subjected to some kinds of enzymatic digestion. So this enzymatic digestion, what it does is that it chops down or breaks down your proteins into smaller fragments called the peptides. So once we have the peptides, so then what we can do is that we separate out the peptides in the liquid chromatography, and then we perform the mass spectrometer. So after the ions are generated, and then what we are doing is that we are identifying the m by c ratios of that, and then finally what we are getting is the peptide mass fingerprints to get analyzed that what are that particular peptide. So once we get the sequence of the peptide, then we can uh, easily try to match it with the proteins, because every protein has certain sequences and certain peptides are unique to that unique to a particular protein. So once we get all the proteins, and once we get all the peptides, so once we can match the peptides or the proteins having two or more than two unique peptides, and once we get that this unique peptides, we have those particular unique peptides, then definitely that belongs to that particular protein, and on the basis of that we can calculate the intensity of that particular peptide as well as intensity of that particular protein. So this is the bottom up approach, where we try to get the identify the peptides and do a database search for the identification of our peptides and then the proteins. So that's why we have started with the peptide selection and the peptide separation. So this is the bottom up proteomics approach that we generally used, or that is being revolutionized the field in identifying a lot number of proteins, whereas the top down proteomics is again advancing currently, and is being the place to where one can explore and to get it rolling as well. Okay, so now I have said many terms. I don't know if you have understood or not, like the MSMS fragmentation and all those for the and the identification of that peptide. So again, what mass spectrometry that we are talking about is a tandem mass spectrometry. So what is the meaning of tandem mass spectrometry? Is that where we are doing a second fragmentation, and for one we are identifying the precursor, and then again we are doing the fragmentation, and then we are finally detecting it. So if you can see here, so this is the sample which is getting ionized through ESI, MALDI, which are different ionization techniques. So ESI, for example, is the one which is being used for different high-end cutting-edge mass spectrometers. So once you ionize using ESI, so you are getting the ions, you are doing the M by Z separations, then again you are doing a collision and you are breaking that fragment and fragmenting it into, then again this fragmentation is going, and then you are finally detecting it. So the basically the major thing is that if you do just one ionization, and once we are getting the result, so that can give us to understand. So if you just think and recall now, so what we said that the mass spectrometers are just measuring the M by Z charges, the M by Z ratios, the mass per charge ratios. So ms1 through ms1, that is after just ionization, which can be M plus H plus or M plus 2H plus on the basis of the charged state. So we can just get the molecular weight of the biomolecule, that is M. So we can get the mass of the peptide. For example, if we are looking for a peptide, so what is happening is that the peptide, for example, if it has a molecular weight of 110 delta or something like that, then not it will be more than that. So if you can think of anything around 1500 delta or 1200 delta something, and if it is getting singly charged, so 1200 plus 1 divided by uh, 1, you can get that it is the 1200 dalton, the peptide mass's fingerprint. But we cannot understand that what is the sequence of that particular peptide, because the molecular weight of the peptide is nothing. We cannot deduce anything, or we cannot conclude anything that this is the particular molecular weight of the peptide, and this belongs to this particular peptide and this particular protein. So we need to have the peptide sequence. So like the in the genome projects, we have to identify the gene sequence. Similarly, for identification and the quantification of the proteins, so we need to have the protein labels and the protein sequences, or to have the peptide sequence for the particular proteins. So that's why this second fragmentation is one thing which requires to help us to identify the particular peptides, and to finally match it with which particular protein it is, and what is the particular sequences of that peptide, and if it contains any unique peptides or not belonging to that particular protein. So this is the way peptide sequences are there, and which happens is that with the creation of the ions, so basically the Y ions occur at the C terminal, and the B ions occur at the N terminal. So if you measure the differences when the M by Z is measured, so the Y3 minus Y2 will give you the, or the B2 minus B1 will give you the monoisotopic mass of that particular amino acid, which if you keep on doing subtraction of the Y ions and the B ions, so then you will get that which particular amino acid does it belong to, or which particular amino acid it corresponds to, and that will give us the sequence of that particular peptide. So for this, for example, of this particular peptide, if we keep on subtracting the Y ions and the B ions, we will specifically, so the major concept here is that the B1 minus B2 and the Y1 minus Y2 will correspond to the same particular amino acid. So once we get the uh, monoisotopic mass of those particular amino acid, then we can deduce the sequence for this particular peptide, and then can the software software can look for that at which this peptide belongs to which particular protein, and can further decide on the can identify help you in identifying that particular protein, which protein is that, and what is its expression level. So that's why these two mass analyzers are required, and that's why have used the terms like the MSMS fragmentation is that after the one ionization MS1 we are getting at the MS2 levels fragmentation, are the what are the different ions to finally conclude that what is the sequence of the peptide. And that's why hybrid configuration is needed to identify the peptides. So the major concept here is that if we are doing just a one fragmentation and doing the one M by Z separation and detection, so we can definitely get the molecular mass of that particular peptide, but we won't be able to deduce the sequence of that particular peptide. So now once we are done, so the basically the whenever in the mass spectrometry based approaches we are whatever we are mentioning, it's not that we just need to identify the peptides or identify the proteins, rather we need to have a quantification. So what is that expression level of the proteins in that particular given particular condition, be it any organism, be it from any cell or in the diseased condition. So what is their expression level? So similarly in the gel based, if you can just recall that in the gel based we have spoken about 2D dyes which gave the quantitative edge. So always we need to have a quantification, then only it will be helpful for us to understand, because simply identification of any particular proteins won't be of any useful until and unless we know that what is expressing more. It is okay that if certain proteins are getting expressed only in a particular condition, and if we can identify those particular proteins that which are getting expressed only during certain condition and not in the uh, other condition, it is fine. But still what we need or what we require is to have the quantification. So now the major there are approaches which are help us to get quantification, to quantify those particular techniques to quantify those particular proteins from the samples. So one is the label based quantification technologies or the quantitation methods. So of which first is the iTrack. So iTrack is something, it's isobaric tag for relative absolute quantification. So the major thing here is to have your samples labeled with certain kinds of tag that will resemble that particular tag will resemble that this is your sample 1, this is your sample 2, this is your sample 3, this is your sample 4. And in this way you can have 8-plex eye track where you can label 8 samples of 2 different conditions you can take, for example control and disease, and that will show that these tags belongs to this particular sample and these tags belongs to this particular sample. And then you can mix, and then finally what you can do is that you can mix and do in the same way you can run in the after you have got the peptides, you can tag your particular peptides belonging to which sample, and then can mix and then can run in your mass spectrometer. So the major thing is that since we have spoken about that the mass spectrometer is something which is just calculating your M by Z charge. So that's why what tag which we are using is having an isobaric tag, so that we are not adding any kind of extra tag to any particular sample. So the all the masses remain the same, so that it does not get into effect in the M by Z charges. So here if you can see the reagents are using one reporter group, one balance group is something which is balancing out, and it is making your group as an isobaric, all the tags as an isobaric ones, so that your sample is not being biased toward having any extra masses or charges which will getting interfered during your separate during your mass spectrometer, and you cannot ultimately compare that it is the difference is due to what. But this group, the reporter group, helps us to what, that one can find out. So if I don't know if it's visible, so you can find out that if the certain expression of that particular peptide or particular peptide is higher in certain samples, so then you will find that the reporter group has to having a higher intensity level, whereas not for the rest of those. So in this way we can get an overview, or you can get an idea of how which peptide is getting higher expression in which sample, and which gives us a way to compare in between two different conditions. So similarly in the TMT is the another approach which works on the same principle, but having giving you an options of having a higher multiplexing options with different levels, but it is also the ones which uses the same isobaric mass tags, which helps you to get and quantify those proteins and those reporters. So for example, you can see after the ms selection happens in the msms quantitation, you can get that the different conditions, different samples having what are the expression of those particular peptides in those particular samples. And then what we can say is that which one is having a higher expression and which one is having a lower expression. So then we can easily get that. So this is the labeled. So this again works on the same principle, and it's so it helps us to having a better multiplexed options. So again, so this is the label based, where we are using uh, the labels to that particular peptides. Again, this is an in vitro. Again, it comes under the in vitro labeling techniques, because the labeling techniques is also another method is present, which gives us an opportunity to have an in vivo labeling technique, or to have in vivo based labeling approach. So this again, the labeling approaches and the technique that one chooses to again remains of each of those techniques have their own pros and cons, like the labeling efficiency, then to have those then how much labeling efficiency is there if all the reporter ions are being getting identified on your instrument. All these things will lead us to to have a quantification technique that will be useful for each for our experiment and for our work. So this is another two label based techniques. One is so once after iTrack and TMT is the SILAC, which is stable isotope level amino acid in specifically for the cell culture, and one is the ICAT, what is the isotope coded affinity tag. So as I mentioned that iTrack and TMT comes under in vitro labeling technique. In vitro means that outside the samples, once the samples have gone, you have extracted out your proteins, you have digested it into peptides, and then you are performing the labeling techniques, and then you are taking it forward. But here in the SILAC technique, what is happening, it is in the in vivo labeling method, where you have cells of your condition 1 and the cells of your condition 2. So what you are doing is that the cells of these two different conditions you are growing in media. So one in one media you are having the cell, so you know that the cells require the medium to grow in culture. So you are using one cell culture with which is having just the normal arginine, but in the cell state 2, which is for example one you are taking as control, and then another you are taking as treated, you are treating the cells with certain drugs. So what you are doing is that you are growing that particular cell in heavy arginine media. So what it will happen if you keep on cell passaging your cells in that particular media, so ultimately that whatever peptides are naturally getting synthesized in your cells functionalities, for maintaining the different functionalities, the proteins that are being synthesized naturally, though the heavy medium arginine will get incorporated into that. So for that, whenever you take out that particular proteins out from that cell and digest it into peptides, so definitely it will be all your heavy level peptides. So these heavy peptides and light peptides, if you compare in the mass spectrometer, so since again you will get the difference that this peptide is belonging to this particular cell state, and this peptide is belonging to that another particular cell state, then definitely you can compare, because one will be having a heavy level peptide and one is a light peptide, or the original, that is the peptide. So you can compare. So now this is an in vivo method, where in the cells, in the cell culture, you are incorporating that heavy medium heavy peptide into your particular cell, where you are culturing it and getting incorporated into it, rather than after you have extracted out the peptide and labeling that. Now, another in vitro labeling method, that is ICAT, is that which is binding to a particular amino acid. Now, the major thing is that in the iTrack and the TMT, it was that the labeling was done on the peptides, but this is being done on the cysteine residues of all the peptides, wherever the cysteine residues are there. So ICAT, what it does is that the thioreactive group will get linked it with the cysteine. So it suffers from major disadvantages, like if you have a cysteine rich protein, it will be the labeling efficiency and all those depends on it, and so that's why it is not so much in use as of now. If you see that mostly one thing that gets very much being used is the iTrack and the TMT ones, not the ICAT ones, due to the those particular reasons. Hello, Ankit. Yes. Yeah. So this is for the in vitro labeling method, which consists of certain thioreactive group and those things to have one uh, purification and to so getting the isotope wooded affinity tag. So these are the few of those techniques which uses different labels to have the quantification and to have quantifying techniques. So now, after these label based techniques are the ones, but have certain difficulties and certain things which becomes difficult. So now the another concept is of for having the label free quantification techniques, where we are not using any kind of labels, not any isotope labels, or any not labeling the amino acid sequences, or not labeling our peptide sequences. Rather, what we are doing is that we are just taking the proteins and digesting it into the peptides and running it into the mass spectrometer, where we are getting the different spectrals and the different intensities. So here, what we can find out, there are two methods through which we can quantify our particular signal intensity. So you can see that where uh, there we were seeing that we got different labels, and with the help of that particular labels or particular those labels, when we are getting our particular peak and those intensities, what we can find out, yes, that definitely there are different techniques, or the different those reporter groups which we can find out and compare in the differences. But in the level free quantification, what is happening, how it is measuring, so this is measuring, one method is that to be inferred by the counting the number of the peptides or the spectra assigned to a given protein. So that method is known as your spectral count method. So since if your sample contains that protein in huge quantity, or in a huge amount, or if it is very much upregulated in your particular expression level, uh, So then what you can see that you are getting that particular peptide, and then that particular will have peptide will have a greater intensity, and so we will have a higher number of spectral counts through which you can identify that this particular sample contains a higher level of amount of peptides expressed for this particular sample. In a similar way, what in the another method you can extract. So if so you know that what we are doing is that we are separating our peptides on a liquid chromatography where peptides are getting eluted at different times. So if peptide, for example, take peptide X in sample 1 and sample 2. So if you have peptide X in both sample 1 and sample 2, then definitely that peptide X you are getting uh, will should, if all your experiment and all your uh, parameters remain the same and is correct, so then that peptide X is expected to release or to get eluted at the same time, for that peptide X to be eluted at the same time for your complete uh, after your complete chromatographic separation at the same time. That peptide X needs to be will be eluting out at the same time. So if that peptide is getting eluted at the same time for both of your samples, that peptide X, then you can do is that you can extract the area of the precursor ions, and we can try to find out the UC curve or the extraction of the single signal intensity. We can measure the area under the curve and can try to find out that which uh, two samples peaks are getting upregulated or downregulated. Now in this level free and after we do the level free quantification technology technique, so there again remains two different approaches or two different methods. Specifically in the mass spectrometry data acquisition is to have the spectra that we are acquiring. So this is on the basis of what? So basis of what means, so in the MSMS quantitation, which we have said in the tandem mass spectrometry, when we have the first MS fragmentation, after the ionization is done and we are selecting out the MYZ spectra. Now, on the basis the next fragmentation that will happen. So the next fragmentation in the tandem mass spectra, that is at the MS2 level, that will be happening. So that will happening in which way? So that leads us to give two different complete different methods of the mass spectrometry. One is the data dependent acquisition, another is the data independent acquisition. So what is the data dependent acquisition? Where the selected precursor or having a very high abundant precursor. So if the intense precursors or having a top abundant precursors, you can say be it 10, be it 20, be it 30. So if those kinds of intense precursor ions are fragmented after your MS1, so then it is known as your data dependent acquisition, where you are fragmenting it after you are getting the most intense precursors. Similarly, but in a data independent acquisition, what we are doing, that you are defining a range, and within that range you are all precursor ions of the defined M by Z range will be fragmented and will be taken forward for analyzing. So here what you are not trying to restrict for fragmenting only the intense precursor ions. So you are not asking your instrument to just fragment that only those particular precursor ions which are very much intense or which were very much high intensity. Rather, you in a predefined given M by Z range, all the precursors will be fragmented and will be analyzed at the MS2 level. So this is all about the was all about the discovery phase or the discovery phase of the proteomics, which we look upon specifically in the mass spectrometry based proteomics, where we get the level based different quantification technologies, including the level based, level free, and the using the method of DDA or DIA. Now we have said that also in the year 2012, which showed that in the year 2012 that it was that the nature method of the paper, which showed that the targeted proteomics can also be used, which in a way, in a very in a high throughput manner, can also be used for validation of certain proteins and peptides. So you can understand now by this time, you should now understand that we are talking about the bottom up proteomics approach. So what in the global phase also what we are doing is that we are getting the peptides, selecting the peptides and identifying the peptides which we are getting further back into the proteins. So now in the targeted proteomics also we can uh, I, select out those particular peptides of those particular proteins which we generally think of, and which we generally see that which gets which needs to be validated, which is coming as a differential regulation and all in the whatever comparison that we are doing. So can be done in mostly in these two ways, which is known as your selected reaction monitoring or multiple reaction monitoring, or the parallel reaction monitoring. So where in the selected reaction monitoring or the MRM, multiple reaction monitoring, which uses as the triple quadruple. So this is nothing, just two mass analyzers which has different properties. So this quadruple or bitrap are all those mass analyzers which helps in the separation of the ions in a different resolution for the better separation of those ions or the fragments that we get, and finally select out those peptides. So in the SRM or MRM, what happens is that there are two sets of quadruples, and in between is the collision cell. So the what we ask, so how it is in the validation phase, or how it helps us in the particular validation of certain peptides and the certain proteins, is that here once we have identified that particular proteins and peptides, and if we want to say that, if we want to check that whether that particular peptide and particular protein is belonging into that sample or not, so then what we are doing is that we are saying that what is the precursor for that particular peptide. Then if it gets fragmented, what is its product. So precursor and that product ion we are also defining and asking the instrument to monitor only that product, precursor product ion, which is known as the transitions. So which helps us to give an idea that this particular peptide or this particular protein is present in that particular sample. So similarly, what happens in the PRM or the parallel reaction monitoring is that we just ask the quadruple or the to have the precursor ion and try to get all the possible fragments that are there. But since the map or the thing that the product and the precursor map for that particular peptide is unique to that particular peptide or for that particular molecule for that reason, so then definitely we get that the we can say that this particular peptide belongs is present in that sample and is has this and we can do a relative quantification to say that this peptide and protein is upregulated or downregulated, and in a way we can try to find out and we can try to validate those proteins without using antibodies in a high throughput manner. So this was all about the mass spectrometry based approaches in the proteomics field. So we have seen that in the proteomics field. So what are the different types of the proteomics that we are there, right from the protein expression, protein profiling, then the interaction. So the major chunk, another role in the functional proteomics or in the interaction getting is to find the how the interactions happens. So here another technique that is being very much used is the protein micro arrays. So this is used for studying different protein antibody, protein interactions, etc. So this has certain different steps that takes into account. One, like the immobilizing your certain agent. So here it is protein microarray. So I'll just give you a brief that the protein microarrays was very much being used and is being used in the lines of the same principle that have been used like for the DNA microarrays. So for the understanding the gene expression, the DNA microarrays has played a huge role where the cDNAs have been, where the mRNAs have been converted into cDNAs, have been using specific deep protection using different photolithography in the glass slide kind of thing, where different probes of those particular oligomers of that particular genes are being placed onto that glass slides, where you can take your sample of interest and can find out that whether from a different cell you can, from a different condition A, you can label your particular gene into one kind of fluorescent labeling, in another you can use another kind of labeling, and then you can try to find out and hybridize those genes in that particular microarray, where you can see that at which place it is getting in hybridized and at what intensity. So then you can understand that your particular genes are getting upregulated or downregulated. So in the same way, protein microarrays were the ones which had been used to find out the having the get to know about the different interactions or the protein interactions. But here the major uh, challenge has been to have the purified proteins to clone and to get the purified proteins out, and then finally add into the glass slides and to fix it in the agent, so and immobilizing it. So but we will see that how it has been done. So here we have shown you one example how these protein microarrays have been used. So here in a disease state, you know that they are not only the different proteins gets differentially regulated, some proteins gets huge upregulated and those things except than the normal level. So this what it does is that it lets you to have a varied kind of or immune reactions happening in a body, and this leads you to the raise of different antibodies. So if you have all the proteins like the 20,000 proteins getting printed on your glass slide, and then if you just using your sample of analyte, be it and serum or be it any sample of your patient sample, if you just spread it on it, so then if that antibody against that particular proteins, so since you know that your proteins are from a normal human being and the antibody should not be present, otherwise there will be an autoimmune reactions. So those particular antibodies, if it gets matched to it and if it gets identified to all those particular proteins, then you will get a signal intensity, the fluorescence intensity, and that you can match and can find out that against which protein which auto antibodies have been generated, and that can be an excellent way of or marker that can be used to identify that which particular proteins and which particular autoantibodies can be used for understanding in the disease progression and disease states. So this is again label based techniques. Now, so in the understanding the protein interaction also, there is our label based techniques and the label free techniques. So whenever we are talking about this getting the intensity out and understanding those expressions, obviously it is we are doing a kind of a fluorescent labeling and those for getting the expression levels. So now this is another very much used technique, which is a label free technique. So where we are not using any kind of label to get any intensity or signal out for that matter. Rather, what we are doing is that we are trying to get in the physical interactions of the change, trying to understand the change in the physical interactions for our understanding that how the interaction levels are, how whether it is having a strong interaction levels or whether not. So this is, for example, this one is known as your surface plasmon resonance or SPR. This is because your this plate is coated with a metal specifically for a gold. So just again to be clear, we have uh, from the identification of the proteins and from getting the proteins level and validating, we have moved on to understand the different interactions. So in this gold medal, if we gold metal plate, if we have our with a thin coat of film, and then if we have the ligands immobilized, then when we do uh, light source on it, so there with the interactions, as the interactions goes on, so there will be a change in the reflectance this angle, and it will show that there is a this refractive index gets changed, and that's why there will be a change in our SPR signal. So SPR is that in that surface plasmon resonance, so it is that at which angle at which the particular thing resonates, and we get a signal out of it, and there is a total in this detector the reflection comes back. So if for example, if we have our ligand on this particular gold chip, and then we are giving the analytes at a certain flow rate, and we are keep on just adding on the flow over this ligand, so to characterize the interaction. So this SPR signal will change with the refractive index, and as the masses will get on increased with the binding event, it will cause a proportional increase in the refractive index. So it will definitely cause keep on causing proportional increase in that particular refractive index. So here you can see that this is the baseline. Then you are keeping on giving the flow the analyte, and if that there is an interaction that is being happening between your analyte and your substrate, that thing that you have immobilized on your substrate, then definitely there will keep on association at the some point of time. At there will be no such left of ligand left where you can the analyte can be bind to it, and then finally if with the increase in the time and in the flow rate, you will have the dissociation, and then finally again regeneration, and then the baseline. So this will give us an understanding to have an to have the association rate, the dissociation rate, and the KD, and this helps in excellent way to understand, to have an interaction between protein drug to proteins, and all these applications we can get an excellent view. So this is a label free technique where we are not making use of any labels. So now another thing is an in a case study wise you can understand of the technique, that is the SPR MS. So we have talked about mass spectrometry mesh technique. We have talked about and seen that there is an SPR or level free technique specifically to have an understanding of our interaction. So now if we need or want to understand that how the kinetic the characterization, and for example the epitope binding we can understand through SPR MS. So what is that? So we are having the samples. One sample we are getting for the SPR. So then we are getting that this epitope is binding. What is the characterization of that particular epitopes against that particular target, and what is the level of the association and the dissociation constants and those things by the sensogram. And in that same epitopes we are channeling it in the electrospray ionization and doing the MS after obviously going through the different steps to identify what is the particular epitopes are there. So this is very much suited for your samples which are having your multiple components, and it allows you to carry out the affinity based isolation of the epitopes. Affinity based isolation can be done in the SPR, where you can get the responses, that how much it is getting binded and how much responses you are getting generated, and then finally identify them. So there are two configurations that are possible. One is your off chip configuration and the other is the one chip configuration. So off chip is this one where you are getting that injecting your sample doing the SPR and then finally ejecting it out and getting the ESI, creating the ions in through the electrospray ionization and then taking into the mass spectrometry. Whereas if you do it in the one chip configuration, then in that same SPR chip, one goes for the multi that ionization, and then goes directly into the mass spectrometer, giving you an one chip configuration where you are not taking it out and doing for the electrospray ionization. So this is in a broader sense for a case study for better understanding. Also gives us an flavor that how different technologies like having completely using the interactions can also help us in identification, and those techniques can be coupled as well for better understanding. So now, once we are understood some and got some feel of the different technologies, so now what is basically the interactomics is? So interactomics is something that we know that the protein interacts with other proteins, but protein also interacts with other biomolecules like the nucleic acids, like lipids, like other uh, agents, and it helps us to have a very comprehensive molecular system. So this is forms a network, or the interactome is a whole complete interactome network which leads to the functionality of another. So one protein can lead to functionalize, can lead to activate of another protein, or can lead to the down regulation of the activation of activity of another protein. So this is the complete network, and the study of this complete network and the interactions is only we can term it as interactomics. So what are it uses? It can help us into determine the roles of uncharacterized proteins. If we can identify certain proteins which have been not very much well characterized through the its interaction pattern, or its through the prediction that how it can be interacted, we can get those kind of ideas. Then it plays a huge role in the regulation of the protein activity, and then the constructing the network of protein interactions. So the different methods of interactions that we can think of about in this particular field is like the transient or the permanent, weak, strong, obligate, non-obligate, homooligomer or the heterooligomer. So these are different methods of interactions that are possible and that can be taken forward. So there are huge significances, so you can understand not only so we just think of networking in our sense. So human beings are very much social organisms, or different animals can be social organisms, but this is not at a macro level, or what we think of like we are trying to connect through different platforms, or to connect uh, in a society, what we try to do to connect. But in a very micro level also, you can see that it remains getting connected, that all proteins, all biomolecules remains to form in a connected, and in a connected process only performs the cellular machinery which makes us running, which makes us uh, alive, and which makes us make all the works done. So the significance of studying these protein-protein interactions is that to understand the cellular processes and functions, to have an idea about the signal transduction, cell growth regulation, protein transport, biomolecular interactions, alteration of the enzyme cascade. So this is a major things, because this can work as a domino effect with when the particular protein, if it gets hampered in its activity, can lead to the dysfunction of a sets of particular proteins, and that leads to a major change in or major challenge in a person to belong in a machinery, in the normal machinery, and it leads to the fault, and it leads to the disease conditions. So different physiological processes, and it is generally governed by this kind of physical interactions, which you can take about, like the electrostatic interactions, hydrophobic interactions, van der Waals interactions, hydrogen bond. These are the some of those interactions through which it is governed. These are the your, you can tell, the physical forces that governs these interactions as a whole, and is making the things possible. So now the comes about, so once we have so if I just summarize you for you all, so we have think about the proteomics field, and to understand and identify different proteins and help in getting those particular proteins, what is their quantification, different quantification techniques. But now if we try to understand the protein protein interactions, again, what are the different approaches that are available. So one is always the conventional approaches, and another is the high throughput approaches. So conventional approaches are like the is to hybrid system, which we will look into some subsequent slide. One is the, again, is the affinity chromatography. So you can, by the name you can understand that to have to extract out something on the basis of an affinity, to just select it out, like the nickel NTA thing, it can be used. Then is the immunoprecipitation. Again, when the term immuno comes, like the antibody, to use antibody to precipitate out certain proteins, so that gives you the immunoprecipitation. Now, what are the high throughput approaches? Like the protein microarrays, which we have talked about, is very much useful, because you can understand that having 20,000 proteins printed on that particular slide, and then you are getting to have an idea that what are the different proteins that have been used, have been differentiated now, is which are getting interacted and which are not getting interacted. Another thing is the level free technologies like SPR or BLI, etc., which are there. So these are all high throughput approaches which we have talked about. But also there are some conventional approaches which are very easy to be used, and uh, which have a very simpler protocol and does not necessarily need a huge cost expensive setup, which we will be looking into it. Okay, so now, before using the conventional method, one is the immunoprecipitation. So immunoprecipitation is one which is being used, which can be used in a very uh, robust way. One is that to have a cell lysates which contains your total protein mixed, which is having different your proteins of interest. Then you are doing an so you know that which particular proteins you are doing, so you are adding that particular antibodies against that, and you will be incubating it. Then antigen antibody will have complex, and you will be now giving protein G. So why you are adding protein A or protein G? Because protein A and protein G has an affinity towards your IgG. So this antibody is your mostly that is being used is the IgG. So this protein A and G will have an affinity towards IgG, and this will pull down. So once you elute it out, so this will pull down your antibody and the protein complexes together. And after you purify those particular antibodies, these particular bits out of it, and then you can run it in gel, run it in Western blot to find out that you have got that same exact protein. You can do in the mass spectrometry, you can do in any of the techniques. So basically, it is depends on your selection of a suitable antibody, then binding of those antibody to protein, and then elution of those antibody and protein complexes, which you are doing by purification and downstream analysis. So now this you can divide the whole process into two conditions. One is for your non-denaturing conditions, because here the antibodies needs to be kept in a very native condition, because to get attached to your protein and to pull out of your protein of particular interest. Whereas in the denaturing conditions, once you have purified it and you are doing the STS page and the mass spectrometry, you are denaturing it. So this belongs to your into the denaturing condition. So now immunoprecipitation has certain merits, like the natural introductions can be identified. You can purify your large complexes, and proteins are in native states, which leads you to have that interactions possible. But the demerits are the specifically the non-specific interactions. If your antibody gets attached to your in a very non in this way very non-specifically way, then what to do. Second major question is that the interactions. So for example, you have an antibody X, and after you have done in the gel, and after done in doing in the mass spectrometry, you have found out that there are 10 proteins. But what so which proteins are getting interacted with which ones? So there is a protein complex. So for example, your antibodies attaching to that particular protein. Now that protein has an interaction with another protein, another protein has an interaction with another protein, and this is a chain that is being made. So now your question is that which proteins are getting interacted with which one has a direct interaction. So this whole complex we have got now to decipher and to understand that this which interaction are the ones that are actually being interacted is one of the major question or major things that remains to answer in the immunoprecipitation technique. So this is the merits and demerits for the immunoprecipitation techniques. So now next is the, but we have another conventional method is the yeast to hybrid system. So what it happens is that it relies on the expression of a transcription factor, like you can take of a reporter gene. So the LAGZ, you can take the LAGZ. So what it is says that it this transcription factor or the reporter gene uh, uh, this reporter gene gets activated by the transcription factor, and the transcription factor has two components. One is your DNA binding domain, and one another is your activation domain. So this works in a kind of a bait and prey system. So if you have a query protein and it is getting so the query protein is fused with your binding domain. So you have a query protein, for example X, which is added to a binding domain, and you have another into activation domain, and the protein that you are querying against means that you are seeing that whether it gets interacted or not is fused with the that which particular protein. For example, you have one protein and it's being attached to the binding domain, and now you are screening 10 proteins to find out that which one gets interact with your particular that protein of interest. So what you are doing is that you are fusing that with the activation domain. So now if these two systems, that both the binding domain and the activation domain is expressed in the yeast nucleus, then the activation or the expression of the gene can only be possible if that both the activator domain and the binding domain gets fused, that is the two proteins interact. So if the two proteins interact, then only you will get the expression of the reporter gene, which you can get through uh, the expression of that, and can choose accordingly that whether which library or among the library which particular protein is interacting with your particular binding domain. So if you have this kind of configuration where you have the genes with binding domain and the activation domain, and these are the query bait, that is the query protein of interest, and the prey which you have an library of proteins that you have, and you are finally this reporter gene will have an expression only when these two gets interacted. So with that expression of this particular gene, you can know that whether the protein have been interacted or not. But the major this can be used in a variety of things, like for the protein RNA. protein DNA, protein protein interactions, and etc., and also can be performed in a quite a cost effective manner without having a very difficult protocol, and can be used to your in large libraries. But the problem is that the localization of the proteins in the nucleus is a must. Then what about if it is we if you haven't noticed that we have said that it is happening in an east nucleus. So if it is not present in an east nucleus, in a non-east environment, what is there? Now this we are saying that it can happen only if the bait and prey system, like in a interaction manner, pairwise interaction manner. So if there is a multi interactions, then what is the case and what is the scenario? So all these questions remains a challenge, and it does not makes the demerit for the is2 hybrid systems. So now comes the in a high throughput technique is the protein microarrays, which is there. So again, the protein microarrays can be of your two types. One is your antibody arrays, and another is your protein arrays. So one way is that to have all the antibodies printed onto a glass slide, and another way is to have all your proteins printed on it. So the major challenge in this kind of setup of this protein microarrays is to purifying the proteins out of the E. coli or any vector system, expression vector system you are doing, and then finally spotting those are the major challenges. But still, in the year 2001, it was shown that around 5,800 clones of the proteins was used for the, and have been spotted, and had tried to find out the interactions, and also gave the interaction with different biomolecules and could be understood. So this was the ones which again boosted the field, and also with the very much achievements of the DNA microarrays, one could the moved the scientists to go ahead in this field of the protein microarrays. So now different types of the protein microarrays which are there. So one is the direct labeling. So what is direct labeling is that the antibodies are printed on your glass slide, that is on the chip of the microarray, and the proteins are getting directly labeled with your dyes that are measuring your fluorescence intensity. So the proteins which you are trying to identify are already labeled, and you are just the with you are flowing your analyte, and where the proteins are getting matched with that antibody, getting interacting with the antibodies, and are giving the expression level. So that is the direct labeling. And another is your sandwich kind of thing, where the same kind of an principle of sandwich ELISA, where you are saying that you have the antibodies printed, you are giving the lysate which you are which is getting matched, and then you are having adding the primary antibody to get attached with your that particular protein, and then secondary antibody. Finally, you are trying to measure the intensity expression level. Another thing is the reverse phase protein array. So these are known as where the antibodies. So just to give you a brief, where the antibodies are printed on the glass slide of the microarray. These are known as your forward phase arrays. Whenever the cell lysates or the lysates or the analytes of interest are printed on a glass slide, so that is known as your reverse phase protein array. So the cell lysate is printed on the glass slide, and the fluorescence is captured through your primary and the secondary antibody. So primary antibody will be added, and then the secondary antibody will be added and will be getting. So that is your reverse phase arrays. Now another array which is used is the peptide arrays, where the different, like the proteins are getting printed, you can have the peptides along with different tags and those things for getting that how much expression levels and those interactions are there. Then are comings are the cell-free. So all till this thing which remains the challenge is to have uh, expressed your purified proteins, and then finally spotting it in on your system, that is on spotting on your glass slide. But after which was becoming very challenge, so then the scientists came up with the thought process and with some innovations where the cell-free expression level of the proteins can be there. That is purified DNA products along with the some requirements for the DNA and RNA to be transcribed and translated to protein, RNA to be transcribed, translated into proteins with certain and certain his tags and certain tags, so that the proteins that are getting synthesized comes back and spreads into the spots, into that particular spots only. So this cell-free expression systems becomes very much useful, like the nucleic acid programmable protein array or the NAPA, which was uh, being invented, so that it all the proteins can be expressed and without using any cell expression, which was becoming very much tedious to tackle, and rather that proteins can be finally analyzed and can be seen for their interaction. Similarly, for the multiple spotting technique, where the PCR products and the DNA was used to directly transcribe and translate into protein and get the interaction. So there was no need for the cells and the cell-based expression and to get the things. So the majorly this thing was again can be divided into two parts. One for the cell-based expression systems or the any expression systems that are there in the cell based expression systems for the microarrays that is being used, that can be talked about as that abundance based arrays, whereas the cell free systems can be mostly talked about as a function based arrays. So this is the uh, major advantage of protein microarrays are that it is very much high throughput with a small amount of analyte or biological specimen or biosamples that can be used and can be analyzed in your particular setup. But the major challenge is that the protein purification remains the major challenge. So these are the different cell-free expression systems to generate microarrays, which is become very much popularized in this field. But the major challenge was to produce structurally and functionally correct protein molecules. So since you are not using any cell system, rather in an in vitro method, you are trying to extract out and try to print out that particular protein. So definitely it takes much challenge to have a structurally and functionally correct protein molecules. So this leads to the accomplishes of the protein system. This could be accomplished by the means of the cell extracts containing all essential animals. So cell extracts can be of your yeast extract or rabbit reticulolysate or different extracts which contains all your information, essential elements for transcription and translation, and some energy sources or modification or certain tags or modifications will help to get the better expression of the systems. So these are some of the cell-free expression systems to generate the microarrays. For example, PISA or the protein in situ array, where it is taking that the PCR DNA and the cell-free lysate, and it is coupled with your transcription. If you can see that it is coupled with transcription, translation, and the protein capture by tag. So the after the protein is getting translated, so it will get in the microarray chip, it will get attached to that particular place only because of the tag, and it will not have any non-specific or not, it will not get attached to different places without which is not defined for that. Then there is a tag capture agent, tagged protein, and the ribosome mRNA, and you can see that it will in the proteins are generated, and you can get you can do the further experiment through it. Similarly, it's in the NAPA, so which is array. You will get the array, avidin, plasmid, and the capture antibody. So the from the plasmid, you are covering the entire array with the cell-free lysate systems, which contains like the anti-GST, the GST tag protein, and ribosome and the cell lysates for extracting it out, and you are getting that particular proteins out of it. So this major thing is that which what is the starting material that you are taking. For example, in PISA protein density array, we are taking the PCR DNA products. Here you are taking the plasmid, and the but the major concept or the major goal is to have the proteins generated outside the cell system, which helps to reduce the problems of having a lot of difficulties in extracting out the proteins and purifying those proteins. And another is like the in situ puromycin capture from the mRNA arrays, which again consists of the modified mRNA and again covering with the entire cell-free lysate and finally getting out the protein. So these are the some of the expression systems which are being used to generate the microarrays. So if we just so this is definitely an overview of the different techniques that are being there, right from the proteomics to the interactomics and studying the interactions. So the major goal here was to understand and have a, uh broad sense and in a flavor to get the whatever happenings in this particular field and what are the different methods that are being, what are the different technologies that are there, and that is helping us to get an overview of the cellular processes. So just to summarize that the proteomics has become an indispensable field in this field of omics after the huge success of the Human Genome Project, but it failed to answer certain questions, and it was the proteomics that is helping us to answer different questions to understand the mechanistic cues and the mechanistic insights of the diseases, as well as in the upcoming era of the personalized medicine and the precision medicine. It is always that the certain to have a understanding that which proteins and which biomolecules are getting uh, will be effective to monitor, to diagnose, as well as the toxicity, to the off target toxicity, etc. Understanding, so that's the major role. So there are different methods, right? The field started from the gel free methods to the antibody based methods or the MS based method. Again, the MS based method, you can see that there are huge techniques and those approaches available. Again, in the then major goal was to understand that the interaction pattern. So interactions are a way, a huge role in understanding the physiological processes and all those particular processes, and that's why we need to have a good understanding. So in the high throughput technique microarrays, and microarrays also the inspired by the DNA microarrays, but in this field of protein microarrays, if we tend to give different proteins, it becomes difficult, and that's why we have a plethora of mechanism through which we can get and we can identify and we can understand how the methods in this field can work upon. So yeah, so this was all from my side as of now. So, and yeah, thank you.